So this video is going to be on tubular secretion. And tubular secretion is just reabsorption in reverse. We're just taking substances from the blood and we're putting it back into the filtrate. And this is going to occur almost completely in the proximal convoluted tubule. And selected substances are going to be moved from those capillaries into the tubule cells and then out into the filtrate. And this is going to be substances like potassium, hydrogen, ammonium, organic acids, bases. And substances synthesized in tubule cells are also secreted, such as bicarbonate. So again, tubular secretion is just taking substances from the blood and putting it into the filtrate. It's going to be important for disposing of substances such as drugs or metabolites that are bound to plasma proteins. So if you recall, when we went through glomerular filtration, we learned that things like proteins are not going to be filtered out. So if something is bound to a plasma protein, it's not going to be filtered during glomerular filtration, so it's going to have to be secreted. This is also going to be important for eliminating undesirable substances that were passively reabsorbed, like urea and uric acid, getting rid of excess potassium, and then controlling blood pH by altering the amounts of hydrogen or bicarbonate in the urine. So for example, when the blood pH drops towards the acidic end of the homeostatic range, the renal tubule cell will actively secrete more hydrogen into the filtrate and retain and generate more bicarbonate, and that's going to help bring the blood pH back up. So let's focus only on the green. So the green is going to be the tubular secretion. We've already talked about tubular reabsorption in another video, but we have this almost exclusively occurring in the proximal convoluted tubule. We have hydrogen, ammonium, some drugs being secreted back here in the proximal convoluted tubule, some urea here in the ascending limb of the nephron loop, and we're going to talk about urea recycling later in this video. We have regulated secretion like potassium in the distal convoluted tubule. This is bioaldosterone, and then also in the collecting ducts as well. One main function of the kidneys is to make any adjustment needed to maintain body fluid osmotic concentration at around 300 milliosmoles. And osmolarity is the number of solute particles in 1 kilogram of water. 1 osmol equals 1 mole of particle per kilogram of water. But body fluids have much smaller amounts, so we actually express this in milliosmoles. And a milliosmol is 0.001 osmol. If the body is dehydrated, the kidneys are going to produce only a small amount of urine. If the body is overhydrated, the body will produce a large amount of dilute urine. And this is going to be accomplished using countercurrent mechanism. And countercurrent mechanism means that there's going to be fluid flows in opposite directions in 2 adjacent segments of the same tube with a hairpin turn. And there are 2 types of countercurrent mechanisms. There is the countercurrent multiplier, and this is the interaction of filtrate flow in the ascending-descending limbs of the nephron loops of the juxtamedullary nephrons, and the countercurrent exchanger, and this involves blood flow in the ascending-descending limbs of the vasa recta. So countercurrent multiplier has to do with the nephron loops. Countercurrent exchanger has to do with the vasa recta. These 2 countercurrent mechanisms are going to work together to establish and maintain a medullary osmotic gradient from the cortex through the medulla. The gradient is going to start at 300 milliosmoles in the cortex, and it's going to go all the way up to 1200 milliosmoles at the bottom of the medulla. The countercurrent multiplier, which remember involves the nephron loop, is going to create the gradient, and the countercurrent exchanger, which is going to involve the vasa recta, is going to preserve the gradient. The collecting ducts can then use the gradient to vary urine concentration. So this slide is showing the concentration increasing as we go from the cortex down into the medulla from 300 all the way up to 1200 milliosmoles. So this is showing the 3 key players that interact with the medullary osmotic gradient. So, and we're going to talk about all this in much more detail. So we have the long nephron loops of the juxtamedullary nephrons are going to create the gradient. So we're looking at these here. This is going to create the gradient. The vasa recta is going to preserve the gradient, and it's going to act as a countercurrent exchanger. And then the collecting ducts are going to use that gradient to adjust urine osmolarity. The countercurrent multiplier involves a nephron loop and is going to depend on the fact that filtrate flows in opposite directions in the ascending-descending limbs. The differences in permeabilities between the descending nephron loop and ascending loop. Remember, the descending loop is permeable to water. The ascending loop is permeable to sodium and chloride. And then it's also going to depend upon the active transport of solutes out of the ascending limb. The nephron loops are not in direct contact with each other, but they are close enough to influence each other's exchanges with surrounding interstitial fluid. So, again, as a reminder, the ascending limb is going to be impermeable to water, and it's selectively permeable to solutes, and salt and chloride are actively reabsorbed in the thick segment. In the descending limb of the nephron loop, it's going to be freely permeable to water and impermeable to solutes. Water is going to pass out of the filtrate and it's going to go into the hyperosmotic medullary interstitial fluid. And as water passes out of the filtrate, it's going to cause the remaining filtrate osmolarity to increase to about 1200 milliosmoles. The countercurrent mechanism. So the more salt the ascending limb actively transports out into the interstitial fluid, the more water diffuses out of the descending limb. The more water that diffuses out of the descending limb, the saltier the filtrate becomes. The ascending limb then uses that salty filtrate to further raise osmolarity of the medullary interstitial fluid. There is a constant, and I'm about to show you a picture of this, and hopefully the picture will make more sense. There is a constant difference of 200 milliosmoles that always exists between the two limbs of the nephron loop and between the ascending limb and interstitial fluid, and the difference is multiplied along the length of the loop. So let's look at this. So first, we have the filtrate going in opposite directions, right? The descending limb is going down, the ascending limb is going back up. In the descending limb, we have water that's leaving. It's permeable to water, but not salt. In the ascending limb, it's going to be permeable to salt, but not water. So let's look at this. This is a positive feedback mechanism. So we have salt leaving the ascending limb here. As salt leaves the ascending limb, the interstitial fluid osmolarity is going to go up. As the interstitial fluid osmolarity goes up, that's going to make water want to leave the descending limb. As water leaves the descending limb, the osmolarity of the filtrate, which is in here, is going to go up. The osmolarity of the filtrate entering the ascending limb is going to be very high. It's going to be about 1200 milliosmoles. And then because of that, salt is going to be pumped out of the ascending limb. And then again, that's going to increase the interstitial fluid osmolarity, which is going to cause more water to want to leave the descending limb. So we have here the fluid at 300 milliosmoles, and it's going to go down into the medulla. As it goes down into the medulla, water is leaving the descending limb, and the concentration, the osmolarity of the filtrate is going up, because water's leaving, right? We have this very concentrated filtrate here at the bend. Then it starts to come back up, and as it goes up, we're going to start reabsorbing salt. And that's going to increase the interstitial fluid osmolarity. And then we finally get all the way back up, I lost my cursor, to here, and we have this filtrate at its most dilute. As it's going to leave the nephron loop, it's going to be about 100 milliosmoles, and this is going to be hypoosmotic to the interstitial fluid. Then we move on to talk about the countercurrent exchanger. And again, this is going to utilize the vasa recta. And the vasa recta are the capillaries that are associated with juxtamedullary nephrons, right? They go around that nephron loop. And so they're highly permeable to both water and solutes. The flow of blood in the vasa recta is also countercurrent. It has a hairpin turn, so blood can exchange salt and water with the surrounding interstitial fluid as it moves through adjacent parallel sections of the gradient. The blood in the vasa recta is going to remain isoosmotic with the surrounding fluid. That means that as the vasa recta goes deeper into the medulla, the osmolarity of the blood is going to go up. So it starts at 300, and when it gets deep into the medulla, it'll be all the way down up to 1200 milliosmoles. The vasa recta is able to reabsorb water and solutes without undoing the osmotic gradient that's created by the countercurrent multiplier. The countercurrent exchanger preserves the medullary gradient by preventing rapid removal of salt from the interstitial space and removing reabsorbed water. The water in the ascending vasa recta comes from the descending vasa recta or is reabsorbed from the nephron loop and collecting duct, which is going to result in an increase in blood volume at the end of the vasa recta. So all this means is that as the vasa recta is moving through, it's going to be picking up water, it's going to be picking up salt, and the volume is going to increase as it moves through. And so it's going to be greater at the end than it is at the beginning. So the vasa recta, again, as it's moving down into the medulla, the osmolarity is going to increase all the way up to 1200. So as it's going down, we're going to be losing water and gaining salt. Continuing, gaining salt, losing water, and this is going to help preserve that gradient that's made by the nephron loop. As it turns back up, it's going to start, salt's going to start to leave, and water is going to enter the vasa recta, and this is going to decrease the osmolarity of the blood, and then that's going to go to the vein. So the established medullary osmotic gradient can now be used to form dilute or concentrated urine. Without this gradient, we would not be able to raise our urine concentration above 300 milliosmoles in order to conserve water. So we want to maintain the blood at 300 milliosmoles. And if we're dehydrated, we want to be able to concentrate that urine and pull that water back in so we can conserve that water. Overhydration is going to produce large volume of dilute urine. ADH production will decrease, and the urine osmolarity will be around 100 milliosmoles. If aldosterone is present, additional ions can also be removed, which will cause the urine to dilute to about 50 milliosmoles. Dehydration is going to produce a small volume of concentrated urine, and maximal ADH is released. The urine concentration can go all the way up to 1200 milliosmoles. With severe dehydration, 99% of water is reabsorbed. So what we have here is a circumstance where the person is overhydrated. If they're overhydrated, ADH is not going to be released from the posterior pituitary. So aquaporins are not going to be put into the collecting duct. And so a water reabsorption is going to go down. And so the person is going to produce a lot of dilute urine. If the individual is dehydrated, then ADH is going to be released from the posterior pituitary. Aquaporins will be put into the collecting ducts. Water will be reabsorbed, and the person will produce a small volume of concentrated urine. Now we talk about urea recycling and the medullary osmotic gradient. So urea helps to form the medullary gradient. Urea enters the filtrate in the ascending thin limb of the nephron loop by facilitated diffusion. Cortical collecting ducts reabsorb water, leaving behind urea. And in the deep medullary region, now highly concentrated urea is going to leave the collecting duct, enter the interstitial fluid of the medulla, and then go back into the ascending thin limb. And this is going to contribute to the high osmolarity in the medulla. Then we have diuretics, and these are chemicals that enhance urinary output, things like ADH inhibitors like alcohol, sodium reabsorption inhibitors like caffeine or drugs for hypertension or edema. These are the questions for this video, and this is all I have for this video.